I first want to thank the organizers from the Parkinson Society British Columbia, or PSBC, for arranging this opportunity for me to share some of the ongoing research done in the Vocalo group at Simon Fraser University. Okay, to give a brief background about myself, my research training as a graduate student was primarily focused in synthetic chemistry, with a little work in molecular biology and biochemistry. And in my research, it was the biology aspect that I became most interested in, which led me to pursue a postdoc position with David Vocalo. Now, since joining the group, I have used my background in synthetic chemistry to build biological tools to study enzymes, and in this regard, also to develop tools to discover new therapeutic agents for Parkinson's disease. So in today's webinar, I'm going to break down my talk into two parts. The first part will discuss some basic research where I use synthetic chemistry to make fluorescence quench probes, and then use these probes for live cell imaging. The second part of my talk, I will talk about some ongoing work within the Vocalo group about using these probes for drug discovery. Before I go into my presentation, I feel the need to mention that the work I will present today is a team effort. In particular, these three colleagues shown here are key members of the group that helped drive this research. To the left is a former colleague of mine who was a postdoctoral fellow, Sami Susioni, who is now a professor at University of Montreal. He helped in the chemical biology aspect. In the middle here is Christina. She's also a postdoctoral fellow and a Michael Smith Foundation and PSBC trainee. She helped in developing the assay for the high throughput screening. And to the gentleman to the right here is a senior graduate student in the group, and he helped in the chemical synthesis of these probes. Alright, now going into the talk, I'm sure most of you are all aware of the two classical hallmarks for Parkinson's disease, or I'll abbreviate as PD. The first is the loss of the dark pigmentation found in the substantia nigra, which is a result of decreased dopamine-producing neurons. To the left here, on the top image, let me just make sure my drawing tool is on. Alright. So to the left here is a patient with Parkinson's disease, and to the right is a control patient that does not have PD. You'll note that in this image, we start to see the loss of the pigmentation found in the Parkinson patient. Now, the second hallmark is the formation of protein aggregates, also known as Lewy body inclusions. These aggregates were later attributed to the protein alpha-synuclein. Now, with the advancements of research and technology, particularly in the area of genome sequencing and bioinformatics, multiple PD-related genes have been discovered. In a previous PSBC webinar by Jordan and Emil from UBC, they did a really nice job in discussing some of the discovery of the genes associated with PD. Now, for those that have not seen the webinar, I encourage you to check out. The link is provided here in this page. One key point I want to mention is that for everybody that does not know, is that each one of these genes encodes for a protein or enzyme that has a specific function within a cell. To give you an idea, here's the figure from a review article published by Cherubini and Wade Martins from the University of Oxford. In this image, we can see some of the genes from the previous slide and kind of get an idea what their function is within the cell. Now, I won't go through all the details with each associated protein, but rather I just want to emphasize how complex PD is. The second point is I want to show or give some rationale to why alpha-synuclein and its aggregates remains to be the most targeted protein as a therapeutic strategy. This is in part because alpha-synuclein aggregates disrupt multiple cellular pathways, and I indicate this in this slide by showing all the alpha-synuclein aggregates that are colored in the orange colored boxes here. To give you just a couple examples, alpha-synuclein aggregates disrupt the ER to Golgi trafficking by affecting the regulator RAB1, which I'll point to here. In addition, alpha-synuclein aggregates also disrupt the chaperone-mediated autophagy pathway. This pathway is important for the breakdown of soluble monomeric forms of alpha-synuclein. Now, we and other research groups, however, decided to focus on the lysosomes and their affiliated protein. The lysosome is shown here. So what are lysosomes? Well, they are the compartment of the cell that's important for the degradation and recycling of unwanted biomaterial. This biomaterial can be glycolipids, proteins or enzymes, and even other cellular compartments. In this figure, I show a few different pathways where the lysosome fuses with the, in this case, the endosome, or in these two cases, with the phagosome. This fusion helps degrade the unwanted biomaterial. Now, because of this, it seems that having a functional lysosome is important for the cell. Now, within the lysosome, there are over 100 different proteins and enzymes that's involved in this breakdown and recycling process. And of particular interest to our group and others is the enzyme encoded by the gene GBA1. This is because in 2009, Ellen Sidransky and co-workers published an article in the New England Journal of Medicine that showed the clinical relevance of the gene GBA1 to Parkinson's. Since its publication and some research thereafter, GBA1 is now recognized as a significant genetic risk factor for Parkinson's. Its role in Parkinson's disease is not quite known, but some researchers have provided some good evidence in its connection to alpha-synuclein. And I will go over a couple of these examples in the next few slides. So in this study, the researchers used a short pin RNA to decrease the GCase levels in the cell. Toward the top left here, we have a Western blot analysis where we can visually see when treating it with a short pin RNA that there was a decrease in the GCase activity, or GCase levels. This in turn led to a decrease in the GCase activity within these cells. Using these same perturbed cells, the researchers showed that there was a twofold increase in the monomeric forms of the alpha-synuclein, shown here. Pardon my drawing. In addition to the monomeric form of the alpha-synuclein, the researchers shows that there is also an increase in the alpha-synuclein oligomers. In another study, Xu et al. Demonstrated that the same phenomenon exists in mouse models when using immunohistochemistry. Now, immunohistochemistry uses a fluorophore-containing antibody that can selectively target alpha-synuclein protein in the brains of Gaucher disease mice. Gaucher disease is a lysosomal storage disease. It's caused by a mutation in the same gene that we're targeting, GBA1, which ultimately leads to dysfunction in the GCase activity. Now, to the left here, or the far left, we have the control mouse, which has normal GCase activity. Oh, sorry, I forgot to mention that GBA1 is a gene that encodes for the protein GCase. Okay, so in the far left here, we have a control mouse that has a normal GCase activity, and it shows significantly less alpha-synuclein levels compared to the Gaucher disease mouse. The increased red signal means there's an increased levels of alpha-synuclein. And in the study, they also show that the same holds true in different parts of the brain, as indicated in the lower images. Now, the figure to the right is an inhibitor-induced mouse model. The inhibitor, CBE, is a small molecule known to block GCase activity. Now, similar to the Gaucher disease mouse model, the treatment of the inhibitor led to an increase in the alpha-synuclein levels when compared to the control. The last example uses Western blot analysis on human patient brain lysates. Analysis of a patient that has typical Parkinson's disease showed significantly increased levels of the monomeric as well as the oligomeric forms of alpha-synuclein when compared to the control to the left here. Patients with type 2 and type 3 Gaucher disease also showed an increased levels of alpha-synuclein and its oligomer, And if we look at these Western blots, they're quite similar to the Parkinson's disease patient. Collectively, these studies showed a connection between GCase activity and alpha-synuclein. Essentially, when GCase activity is reduced, there is an increase in the alpha-synuclein and its oligomers. Now, while not conclusive, this may aid in the development of Parkinson's disease. Because of this, if we can increase GCase activity, we may be able to prevent the accumulation of the alpha-synuclein and its oligomers, which can potentially prevent or delay the onset of Parkinson's disease. Now, some research groups have in fact already demonstrated this in Gaucher disease mouse models using gene therapy or pharmacological chaperone therapy, and those references are shown here. Now, in the Vocalo group, we want to try to unravel the connection between GCase and alpha-synuclein. However, for us to do this, we first need to know the GCase's function. So GCase is a lysosomal glycoside hydrolase that processes glucocerebrosidase, or GC, the structure is shown here. GCase's main function is to remove the glucose residue that's shown in red to produce the lipid ceramide. An important concept for us to know is, or to be able to study GCase, is how can we monitor this process inside cells? The common approach actually does not measure GCase activity directly in cells, but rather in its cell lysate. By this I mean this process of measuring activity in cell lysates involves first growing up your cells. You would then plate them, and when you plate them, you would then treat them accordingly, say with a small molecule or a gene therapy as I described earlier. After a short period of incubation, we will then kill these cells by lysing them, and then we will use the external probe to measure the enzyme's activity. What we want to do, however, we want to be able to measure GCase activity directly in live cells. And we can do this if we have a probe that enables us to skip the whole cell lysis step. And this can be done by using fluorescence microscopy. As an example, towards the bottom here to the right, here's an example of where the intensity of the green color here could correlate directly with the enzyme activity, where the more green color you observe will mean the higher enzyme activity, while the less color would indicate lower activity. Some benefits of using a probe to measure GCase activity directly in live cells is that you will be able to get a direct readout without the dilution or cell lysis step, and the observed activity will be enzyme's native cellular environment in the presence of potential GCase modulators and pretending binding partners. Now, in developing such probes, Vocalo Group has some experience in this area. The probe design that we focus most of our attention on are fluorescence quench probes. As the name implies, this probe contains a fluorophore and a quencher. When the fluorophore and quencher is in close proximity to each other, the quencher will essentially absorb all the energy or light that's produced from the fluorophore. When the probe, however, is processed by the targeted enzyme, it separates the quencher from the fluorophore, thereby producing a fluorescent signal that's measurable. Some benefits of these types of probes is that these are dark to light fluorescence imaging, meaning that any fluorescent color you see will indicate some enzyme activity. These compounds are sensitive for detection. They also have tunable wavelengths. By this I mean we can use different colored fluorophores, and I'll give reason to why that's important later. And these probes also have good stability. Now, using the fluorescent quench probe concept, the Vocalo Group had developed a probe back in 2015 to target GCase. Now, in this design, the fluorophore was attached to the A-glycone portion, shown here, and the quencher was attached to the portion in which the enzyme cleaves. So once this probe comes in contact with GCase, it is processed, separating the fluorophore and the quencher, and then giving the red fluorescent signal shown here. To the bottom here is the live cell imaging experiment, where when the probe comes in contact with GCase, we see the red fluorescence color for the imaging. Now, a caveat to this design is that the probe had poor solubility in aqueous conditions, and it was processed by the GCase extremely slowly. And therefore, we worked to try to improve the design of these fluorescent quench probes. We made some initial attempts at trying to improve the solubility of the probe by adding a sulfonate group. However, as the red X indicates, this probe did not work. We also tried to improve the turnover of the probe by changing the structure that's shown in blue to the right. These changes, however, did not work to improve the probe also. We decided to change the design of the probe entirely by using a design a colleague had developed back in the late 2017. To the left is the original design of the probe that I showed you previously, and to the right is the new probe design. This design puts the fluorophore and quencher together on the part that which the enzyme cleaves. Now, to be able to test this probe, we first need to do some synthetic chemistry. And as a synthetic chemist, I felt the need to show at least one slide to show some of the chemistry used to develop some of these probes. Now, I won't bore you with all the details here, but I do want to point out the last steps of the synthesis to give you a visual of what these probes look like in solution. The second to the last step involves attachment of the fluorophore, known as BODIPI. After purification and in solution, we'll note that the color of the fluorophore, or the color of the probe, is this bright green fluorescence. Now, attachment of the quencher, however, and the final step, as I mentioned before, the quencher will essentially absorb all the light that the fluorophore gives off. And you'll see in this image here that the probe is actually this black color instead of the bright green fluorescence. And when you treat this probe with GCase, it essentially will give that green fluorescence again. Now, with this probe in hand, we tested to see if it works. Unfortunately, this probe did not work once again. We rationalized that if we can remove this oxygen and this methyl group here, we can improve the probe, and indeed it did work with this probe in the middle. And the last probe design, we did some slight modification where we switched the fluorophore and the quencher positions, as well as we used a different fluorophore, and I'll give reason to that shortly. So taking this probe in the middle, we tested the probe inside live cells. Excuse my button got stuck here. Unfortunately, this probe did not behave the way we wanted to. I actually had a nice video here to show, but we had some issues getting this video to play using the software, so I had to resort to using some still images for visual aid. So here's our imaging results shown here. What we'll see is that the green color is actually shown everywhere, and this is not ideal. Ideally, when a probe is processed by the targeted enzyme, we want the fluorophore to enter the cell, go to the specific cellular compartment, the lysosome in this case, and when it's processed by the enzyme, the fluorescence will remain in that cellular compartment. But in our hands, this probe, basically the fluorophore had diffused outside the cell completely, showing that green color everywhere. Now, to solve this, as I mentioned, we made that last probe where we simply switched the fluorophore and the quencher position and changed the fluorophore itself. Once again, I had a video here. We can show the video. In this image, I just want to point out that the nucleus of the cells was stained in blue, as shown here. And then over a short period of time, the red color you see is our probe being processed by GCase. And the difference between these images is that the red color you see is much more order. And because of this order, this suggests that the fluorophore is not actually diffusing outside the lysosome. Now, to ensure this, we performed colocalization experiments. Once again, the blue indicates the nucleus of the cell. In the middle here, we have we use a dye called lysotracker green. This dye is known to specifically target the lysosome of the cell. And the last bit is our probe that is being processed by GCase. The large image to the right here is a merged image of the three shown to the left. And what I just want to point out is that there is good overlay between the green and the red color that we see. This overlay provides good evidence for localization of the fluorophore coming directly from the lysosome. Now, to ensure that we are in fact targeting the proper enzyme, we use an inhibitor that is known to selectively block GCase activity. Essentially, in this image, what we see is there's a significant decrease in the red color you see, shown here to the bottom left. This provides good evidence that our probe targets GCase specifically inside the cell. Now, in summary of this work, for the first part of my talk, we managed to develop a new and better probe that is capable of monitoring GCase activity within live cells. We hope this probe will help other researchers studying GCase and its activity, and hopefully those research groups will be targeting GCase as a therapeutic target for Parkinson's disease. I did want to mention that we managed to develop an even better probe that is almost 100-fold brighter than the first original probe. And I mention this because in the second part of my talk, you will see at one point when we switch from the red fluorescence to the green fluorescence in a screening campaign. And that's because this particular probe was developed in that time period, and we decided to use it instead. So in the second part of my webinar, I will discuss how we use these developed probes in a high-throughput screen for drug discovery. So in general, there are two approaches for drug discovery. In either approach, we need to know the targeted enzyme, which is GCase in this case, and we need a probe to measure enzyme activity. Now, the first approach involves rational design of small molecules. These small molecules tend to be structurally similar to the substrate the enzyme is responsible for processing. In this case, it's this substrate called glucocerebrosidase, as I showed you earlier. And as an example through rational design, to the right here, I show two compounds that have been explored as pharmacological chaperones for treating Gaucher's disease. And in these structures, we can see they're actually somewhat structurally similar to the substrate that the GCase targets. The second approach undertakes a screening campaign where you generally have a large compound library, likely something greater than 5,000 compounds or so. The structure of these compounds are known, however, you likely will not have any knowledge of whether these compounds will work or what these compounds will even do. In our approach, we focus on the latter. In this approach, the drug discovery process is summarized here. There are multiple parts and phases that lead to the successful development and approval of a drug to treat a disorder or disease. The first part involves basic research, commonly done at academic institutes. This work can identify proteins and enzymes that's important for a specific disease or function, as well as provide some tools to study these proteins or enzymes. And these tools could be inhibitors, or they can be probes to monitor the enzyme's activity. The second phase is the drug discovery phase. This most often involves a screening campaign to find potential compounds for your purpose. The third part is preclinical step, which involves careful evaluation of the compounds that show desirable results in human tissue. And you would then measure the efficacy and safety in animals. And lastly, the clinical studies, which is involved in human testing before it's approved by the FDA and cleared for market. You'll note that in this whole process, the number of compounds shown here taken forward to each step will dramatically decrease, to which towards the end only one of these compounds will be approved. Now I just want to point out that we are not clinicians, so our work is primarily focused on the first three steps shown here to the left. So in basic research, we can separate this into three sections. For target identification, we already know we're going to be targeting the enzyme GCase. For probe development, in the first part of my talk, I already talked about discussing and developing a probe to target GCase inside live cells. The last part is developing an assay that is suitable for high-throughput screening. Now, in most cases, screens are performed in vivo. By this I mean outside the protein or enzyme's native cellular environment. This is primarily done because this method is significantly easier as you only have one protein or enzyme to deal with. In our screen, however, we were dealing with live cells. This becomes significantly more complex as within a single cell there are an estimated of over 20,000 proteins and enzymes. We have to deal with the plasma membrane, and we also have to focus on enzymes that's in our specific cellular compartment. While this becomes more complex, the obtained data, however, will be much more useful, and it could also enable you to discover new therapeutic targets. After several rounds of testing and optimization, we were able to, for the first time, demonstrate that our probe and assay was suitable for high-throughput screen using a 384 well plate. This is an example of a plate that we ran through our screen. Toward the top right of this plate, that's shown in this red box, we show that there's primary controls to the left. We have compounds tested that's also incubating in the presence of our probe, and finally we have a secondary control where we can inhibit the enzyme activity. So now that we have our probe, our cells, and the assay developed, we are now ready to begin our screen. Our screen consists of 70,000 compounds that's placed over 214 384 well plates. Now the general workflow of our high-throughput screen is shown here. It involves first dispensing the cells. We will do incubation of the cells. We will then treat these cells with our compound library, incubate it for another 72 hours, add our probe that we developed, incubate it for 1 hour, wash the cells, and then add in imaging media as well as a fluorophore to stain the nucleus of the cell. And lastly, we will perform our imaging and perform data analysis to obtain the results. Now, this step involves, while there are multiple steps involved for every single one of these compounds, this process is all automated by a recently developed high-throughput screening facility at Simon Fraser University. I actually had some nice videos to share with you all as well, but again, I can show the video, but I encourage everyone to check out this link to see the high-throughput screening facility in action. One thing I admire is the dispensing and pipetting of liquids. I think is by far the coolest part of the video in my opinion. I might be a bit biased because I know that this whole process is the most time-saving feature of the screening facility for anyone that performs assays. So in our screen, what we are looking for are two types of compounds. One are inhibitors that block GCase function. In this example, we'll see there's a decrease in the green fluorescence color that we see. While this may seem counterintuitive, there are a number of studies that demonstrate that inhibitors can act as pharmacological chaperones when used at the appropriate concentration. Therefore, these compounds could be explored as a potential therapeutic agent. The other type of compounds are known as activators, where towards the bottom here, we would see an increase in the green fluorescence color you see, suggesting that there's an increase in the GCase activity. Now, in our screening campaign, we managed to screen 208 of the 214 plates. We experienced a couple of screening hiccups along the way. Analysis of the current data is still ongoing, but I do want to share some data from about 20,000 compounds. In this data set of the 20,000 compounds, we managed to find 214 compounds that acted as activators. These candidate hits, or a couple of these candidate hits, can be shown here. Toward the top here is a control where the cells are treated just with our probe. And towards the bottom are 3 images with 3 different compounds that showed a fluorescent increase and presumably an increase in GCase activity. Towards the bottom right of each image, you can see there is quantitative data of each one of these compounds of how well they actually influence the fluorescence, increase in fluorescence. These candidate hits could serve as new potential leads for Parkinson's therapy. Now, there still remains a significant amount of work to be done on our screen. We currently need to finish analyzing the data and validate those candidate hits. The hits that showed an increase in activity will thereafter be evaluated by a secondary screen, or secondary in vitro screen shown here. If the compounds show an increase in GCase activity in this in vitro screen, these compounds will be optimized to potentially increase its potency. To the right, if the compound does not show any activity, but it inhibits GCase activity, we could potentially explore these compounds as pharmacological chaperones. And finally, if there is no increase in GCase activity and there's no inhibition, this could potentially lead to discovery of new therapeutic targets that increase GCase activity. So in summary, I hope our research can lead to the discovery of new compounds and or the discovery of new protein targets for PD therapy. I realize the webinar is a bit heavy on the science, but I hope to at least provide some new information about Parkinson's, as well as share some of the ongoing work that's being conducted to target PD, in particular with our group targeting GCase as a therapeutic target. For acknowledgments, I first and foremost need to thank my supervisor, David Vocalo, for funding the work and being an awesome mentor, as well as current and past group members. The group members in red, I already mentioned earlier, are key players in the group that help get the project growing, and the members in blue are ones that have contributed to some aspect of the project. I also need to thank the funding agencies, in particular Michael Smith Foundation and Parkinson Society British Columbia, for funding a postdoctoral fellowship. And lastly, just some general information. I am not a clinician, however, the general consensus for PD prevention are exercise, a healthy diet, and sleep. For those that want to help, PSBC can connect you to options available for participation. A link is provided here. In addition, the Center for Applied Neurogenetics also has options available for participation. Their link is shown here. The Vocalo group is also seeking blood donors to test a new method for measuring GCase activity in peripheral blood mononuclear cells. You can contact me. My email address is shown here. I can share some information and direct you to a person to participate. And lastly, as a researcher, I'm always keen to share some new research. One area that is a bit outside of my scope of research, but I find fascinating, is some ongoing work about ketogenic diet and intermittent fasting for the prevention of PD. For those that's interested, I share 2 reliable resources towards the bottom here. I encourage you to check out. And thank you for attending this webinar, and I will be happy to take any questions.